All right, so hello everybody that are here live and on YouTube. So today Iris and I are going over topic 7, which is the first week of cell cycle. So we have this week, which is kind of like the introduction going through G1, and then we have next week. So just further looking at it a little bit more in like an overview, right? So this week we're going to focus on regulatory mechanisms within G1, right? And next week, which is topic 8, we're going to be focusing on regulatory mechanisms in S, G2, and M phase, right? As well as important cell checkpoints in these phases. So make sure you have like a good grasp for this week's material because it definitely builds for next week as well. And while you're listening to this review and also like watching Laura's videos for this week and next week, just notice how a lot of these regulatory mechanisms we learn about work around DNA, and maybe make some like assumptions of why you might think that's important. Okay. So, all right. So this is kind of the overview of the cell cycle, right? So we have these 4 distinct phases. So I'll start up here with G0. So G0 we define as like a non-dividing state, right? So usually the cell just went through the cell cycle and the cell's waiting for a signal or growth factor signal to actually enter G1. So once the cell receives that growth factor signal, the cell is going to be in G1 phase. We call this gap or the first growth phase, right? And in G1, the cell's can prepare for DNA replication. It's going to check to make sure it has enough nutrients, the correct cell machinery, and that we have no mutations within our DNA, right? Also in G1, the cell is going to commit to the cell cycle, and this is after we pass that restriction point that's over here at the end of G1. After G1, we're going to bring it into S phase. So S phase is defined as when we start DNA replication. And in S phase, we also have end of phase checkpoint, which you'll learn about next week. After S phase, we'll go into our G2 phase, which is our second gap or growth phase, right? So in G2, the cells can prepare for chromosome segregation, and we also have an end phase checkpoint. And then finally, after G2, our last phase, M phase. So M phase is where the chromosomes are actually going to segregate from each other, right? And we're going to go through that PMAT, right? Prophase, metaphase, anaphase, and telophase. We also are going to learn about an important checkpoint next week called the metaphase checkpoint. So just a couple of reminders here. Remember, interphase is G0, G1, S, G2, and M phase is the mitotic phase, right? And a couple other points here, remember all cells are metacells, and also keep in mind that cells control when they divide because cell division requires a lot of nutrients, and the cell doesn't want to waste any of these, doesn't want to waste, time and energy going through the cell cycle if it's not going to be successful, right? So these first slides are going to just go a little bit more in detail into each of these sections, right? So like I said before, G1 is the beginning of the cell cycle, right? We call it the entry point into the cell cycle. So most eukaryotic cells need to receive a growth signal to start thinking about cell division. So we learned the RTK RAS MAPK pathway, I think like either a couple weeks ago. So this is our growth factor signaling cascade for cells to actually enter into the cell cycle, right? So also in G1, remember that cells are going to check for DNA damage. They're going to make sure that the correct size, they have enough energy, and also that they have the nutrients available to actually go through the cell cycle. So, right, this would be like carbon and nitrogen. So if these preconditions, as I stated earlier, were met, the cell is going to pass through the restriction point, right? So let me just move this up a little bit. So the restriction point is going to be like around like right here. And the reason why I pointed out, because remember, once we pass the restriction point, we're still in G1 phase. So just keep that in mind, right? So I also have down here restriction point. It's also called R, and in yeast models, they call it start. So there's a couple different names, right? But remember, after we pass the restriction point, this is the point of no return. So the cells have 2 main options after this point. Either finish the cell cycle or apoptosis, which is like cell death, right? Is there any questions on G1? All right, so the next phase after G1 is S phase, right? So in 285, so this point is it's a little bit fuzzy, I guess, with who you talk to or the papers you read, but in 285, right, the class you're in right now, we define S phase as when the cell begins to replicate its DNA, right? So in S phase, we have the S phase checkpoint, which is, like I said before, it's kind of fuzzy. So sometimes it's not discussed in textbooks, but Laura talks about it. So I thought it would be important to bring it up. So the S phase checkpoint, which is when cells can temporarily halt DNA replication if an error is detected, and then resume DNA replication or perform programmed cell death, like as we stated in the previous slide. And we'll talk more about DNA replication or repair in topics 9 and 10. So after S phase, we have G2, right? And remember, G2, cell's going to check to make sure DNA is done being replicated, that there's no DNA damage from replication. And the cell is going to continue to grow, right? It's going to double its proteins and organelles. You know, if we create two daughter cells from mitosis, you know, we can't just take a cell's normal, you know, organelle count and split it in half. The cell wouldn't be able to survive that way. So we have to actually double these proteins and double these organelles. In G2 phase, we're also going to get the G2 checkpoint, which prevents cells from entering M phase to make sure that the cells are the correct size and that they thoroughly check their DNA damage for DNA damage. So, and like I said, the final phase, M phase, right? So during M phase, spindle assembly and chromosome segregation can occur. So we're going to go through that prophase, metaphase, anaphase, and telophase and cytokinesis in M phase. So in M phase, we have the spindle assembly checkpoint, also called SAC, and which is also called the metaphase checkpoint, right? And this can prevent the cell from entering anaphase. So when we're actually pulling the chromosomes apart, and it's going to check to make sure we have proper microtubule attachment, right? Because if you don't have proper attachment, we're going to get ploidy problems, which is, you know, cells getting a different amount of chromosomes from what they're supposed to have, right? So that's the basic overview of what I think you should know about each phase of the cell cycle. So now we're going to get a little bit into the cell cycle control system, right? So remember, the cell cycle control system is regulated by cyclically activating and deactivating protein complexes required for initiation or regulation of DNA replication, you know, chromosome segregation and cytokinesis, right? So cyclically meaning as in like a circle, right? It's constantly happening and shutting off or turning on, you know, so on and so forth, right? So this is done by what we call kinases and phosphatases, right? So we learned about kinases earlier in the semester, right? They phosphorylate proteins. So they take, you know, a phosphate group and add it on to an R group of a specific amino acid, right? And we also have phosphatases, right? So they dephosphorylate proteins. They actually remove that phosphate group. And a key point here that Iris will talk about later is that phosphorylating a protein doesn't always mean activating it, right? So an example of this is WEE1, which we'll talk about in like a couple slides. And just the last point here, remember our main kinase is actually doing a lot of these functions is CDK, right? Cyclin dependent kinase. Is there any questions on any of that? All right, awesome. So let's talk a little bit more about this cyclin dependent kinase, right? So CDK, Laura calls it the CEO, right? Because it kind of controls everything, right? Everything. The way the cell can progress through the cell cycle is completely controlled through CDK, right? So we call it the master cell regulator, right? So CDK is what we call a serine threonine kinase. And what that means is that CDK with cyclin, right, which we'll talk about in a little bit, is going to bind ATP, and it's going to take that third phosphate on ATP and attach it to the hydroxyl groups on serine and threonine amino acids, right? So it's going to take this third phosphate, it's going to cut it in half, and it's going to add it to the hydroxyl group, right? Connect it right to the oxygen. So we have a couple different ways we can regulate CDK, right? We can regulate CDK through cyclin levels, right? Through transcription of cyclin and the amount of cyclin within the specific stage of the cell cycle, right? We can also regulate CDK through phosphorylation, which we'll talk about through CDK activating kinase, right? And we can also have the phosphorylation in reverse through WEE1, which actually deactivates CDK. So another way we can regulate CDK is like CDK inhibitors, which I think there's a slide in here about P53, right? So CDK inhibitors kind of inhibit the CDK cyclin complex. And you're usually going to see it in G1 or within response to DNA damage throughout the cell cycle, right? So in general, we have two main ways to activate CDK, right? So CDK first has to bind its partner cyclin, right? Because it is a cyclin-dependent kinase, and then has to be phosphorylated by CAK, right? CDK activated kinase, right? And keep in mind, at any time you reach a checkpoint, right, the cell brakes are put on by inhibiting or deactivating CDK, right? So if the cell is not ready to go from G1 to S phase, you actually have to inhibit CDK, right? Or CDK is going to keep on doing its job. Any questions on that? All right, so let's talk a little bit more about cyclins, right? So like I said before, cyclin are required for CDK's kinase activity, right? So they activate CDK when they bind together. And it also tells CDK what to phosphorylate, right? So it gives binding specificity, right? So CDK is going to bind to cyclin, and then cyclin and CDK as a complex are going to make an interaction with its downstream target protein, right? So they both contribute binding interaction. That's why CDK is dependent on cyclin, right? So cyclin regulation is a little bit different. It's more regulated on like the transcriptional and translational control. So if we need cyclins for, like a specific cyclin for S phase, transcription factors are going to increase the regulation or increase the transcription of cyclin, right? Which are eventually going to get translated into a protein, right? So it's kind of controlled on the transcription translation way, right? And then when we don't need a cyclin anymore, so for example, if we go from G1, we needed, cyclin, I'm just making this up, like let's say in G1, we needed cyclin, E, right? But in, S phase, we need cyclin F, right? So we're gonna have ubiquitination of cyclin E because we no longer need it, right? So ubiquitination is kind of just, it's kind of like a cascade of like these E1, E2, and E3 complexes. But I think for this class, all you should know is that we have cyclin, ubiquitin is added to it, it's going to get destroyed in the proteasome, right? It's going to get targeted and it's going to go to the proteasome to be recycled, right? Those amino acids are going to be recycled. So that's how cyclin is regulated. So kind of just going into how we actually activate this CDK cyclin complex, right? So we have inactive CDK, as you can see here. Cyclin is going to bind to it. It's going to pull back this T loop in CDK, right? And when it pulls back this T loop, it's going to reveal an ATP binding site, right? So now ATP can come in and cyclin CDK complex can actually take that third phosphate group and target, like we said earlier, serine and threonine R groups on target proteins, right? So cyclin pulls that T loop back and allows ATP to diffuse in so that this complex can phosphorylate downstream protein cyclically through the cell cycle, right? And remember, CDK activating kinase, which we'll talk about in a couple slides, also phosphorylates in the T loop to further stabilize this conformation. Any questions on this? All right, so now let's talk about CDK and cyclin as a whole or as a complex, right? So remember, CDK functions throughout the cell cycle with different cyclin partners. So for the most part, we would say that CDK levels stay the same throughout the cell cycle, right? And so this brings up a question of like, how do you get the same CDK functioning through the cell cycle and targeting different proteins, right? And that's where cyclin comes in, right? We have a specific cyclin for each phase of the cell cycle usually, right? So like I said earlier, both CDK and cyclin are going to contribute binding interactions. We get specificity for downstream target proteins. And like I said before, cyclin levels are going to rise and fall throughout the cell cycle, as you can see from this schematic. Cyclin S, M phase cyclins, and where CDK concentration would kind of just stay constant, right? So it's always there. It's cyclin that's actually going to allow us to have specificity within the cell cycle. So is there any questions on this part? All right, so this is an example of a CDK inhibitor, right? So this pathway is using P53, right, which is a transcription factor. So for all of you guys that might be a little rusty on what a transcription factor does, it's going to bind to the promoter of a gene, and it's going to help recruit RNA polymerase to actually transcribe and then later that mRNA be translated into a protein. So how this P53 pathway works is we have DNA damage, right? We have DNA damage in our cell. This is going to get, this is going to get detected by some pathway, a kinase cascade pathway. I'm not too sure what it is. And it's going to phosphorylate P53, right? And when it phosphorylates P53, it's going to keep it stable, right? It's going to be a stable activated version of P53. And what this P53, what this stable P53 conformation is going to do is, like I said, is a transcription factor. So it's going to bind to the promoter of a gene. And this gene is what we call P21, right? So it's going to bind to the promoter of the gene that goes for P21. It's going to recruit RNA polymerase. RNA polymerase is then going to transcribe P21's mRNA. A ribosome is going to come on, pick it up, and it's going to translate it into this P21 protein, which is actually a CDK inhibitor protein. And it's going to bind to active CDK. And it's going to bind to CDK and cyclin. And it's going to inhibit it, right, until the cell fixes its DNA. And then the cell can go back, finishing up the steps of the cell cycle. And I have here, so if DNA damage is irreversible, P23 can also signal for apoptosis. So is there any questions on this? All right, so have Iris do this part. So I'll stop sharing.
All right, I'll share my screen.
I think it just the slideshow.
Sorry.
Oh, I said, yeah, I think it's just the slideshow button for.
All right, perfect. All right, now we're going to talk about mechanisms of control and regulation in G1. We're going to start with just a list of like everything that happens during G1, and then we're going to talk about some of them specifically. So cells start in G0, that's the non-dividing stage. If they're in G0, they're just gonna function normally. They're not growing, dividing, anything like that. When they receive a growth hormone signal, this is often through the RTK, RAS, MAPK pathway, that signals them to enter G1, and if everything goes like smoothly, they'll be able to divide. The next thing that happens in G1 is that the cells will check for DNA damage, make sure it has enough nutrients, and it's just also time for the cell to grow. And then early in G1, the pre-RC will form. That's the complex that we'll talk about a little bit later in the slideshow. And it's important for the cyclin CDK levels to be low for that, because high levels of CDK will inhibit the formation of the pre-RC. Next is full activation of CDK. We'll also talk more about that. And then that will allow the cell to pass through the restriction point. That involves E2F and RB. Those are two proteins that we'll also talk more about. Next, proteins are added to the pre-RC to form the pre-IC. And from there, the cells can start replicating their DNA and enter S phase. Remember that S phase is defined in this class as the moment that cells begin replicating their DNA. Okay, so the first thing on this list that we're going to go into a little bit more detail on is going to be pre-RC formation. So pre-RC is the pre-replication complex. It starts when the ORC or ORC or origin recognition complex binds to the DNA. I think that's actually the next slide. But as I said before, the pre-replication complex can only form when CDK levels are low, which only happens at the beginning of G1. So the high levels of CDK will inhibit the complex from forming, which is important also for later on in the cell cycle when CDK levels are high, because it would be bad to have a pre-replication complex be forming some other time during the cell cycle. So it's only helpful if it is formed early in G1. Okay, so as I was starting to say before, the ORC or the origin recognition complex binds to the DNA. It's an ATPase, and it helps recruit a protein called CDC6, which is also an ATPase. CDC6 recruits another protein called CDT1. And CDT1 is not part of the pre-RC, but it does help recruit the next protein. So that's why it's important. So it recruits MCM 2 to 7. It's just, MCM is just helicase, which is also an ATPase. And then CDT1 dissociates, it leaves. So it's not a part of the final pre-RC complex. And once the MCM is loaded, that's the end of the pre-RC complex, and that's called origin licensing. So origin licensing is when the MCM gets loaded onto the complex and the complex is complete. Okay, now we're going to talk a little bit more in detail about full activation of CDK. So CDK levels are always present. Cyclin levels are what's more regulated. Cyclin starts being produced as soon as the cell receives a growth signal. So once cyclin binds to CDK over here, it's still a CDK cyclin complex, but it doesn't mean that the CDK is active if it's bound to cyclin. Once cyclin CDK bind, there's an inhibitory kinase called WEE1 that adds an inhibitory phosphate. So it phosphorylates CDK, but it's an inhibitory phosphate, which means that it causes CDK to be inactive. Another kinase that phosphorylates CDK is called CAC, CDK activating kinase, and it adds an activating phosphate. But as long as the inhibitory phosphate is still on CDK, CDK will not be active. So basically, if we want a cyclin CDK complex to be active, we have to cleave the inhibitory phosphate. And we do that with CDC25. So CDC25 is an activating phosphatase. So phosphatase means that it takes off a phosphate. Activating means that it causes CDK to be active. So here is what we saw on the last slide. It's the CDK with the cyclin. It has its two phosphates. One is inhibitory and one is activating. And since the inhibitory phosphate is on CDK, it means that CDK is inactive. It doesn't matter that the activating phosphate is there. So CDC 25, as we said, is an activating phosphatase. It takes the phosphate group off of CDK, and now you're left with active CDK because it no longer has the inhibitory phosphate over here. It just has the activating phosphate, and now it is active. All right, now we're going to go a little bit more in detail about the restriction point, E2F, RB, all that fun stuff. So this happens right after the CDK becomes active. Now that the CDK is active, it can phosphorylate a protein called RB. And RB is bound to E2F in a way that holds E2F inactive. But once CDK is active, it can phosphorylate RB, and that causes RB to let go of E2F. And E2F is a transcription factor that will upregulate transcription of proteins that are needed for S phase. So once E2F is active, it will allow the protein to, it will allow the cell to continue to S phase because it'll help make the proteins that are necessary for S phase. And as soon as RB lets go of E2F, that means that the cell has passed the restriction point. Let me make sure I didn't miss anything. Okay. So now we'll talk about proteins that are added to the pre-RC to form the pre-IC. All right, so we formed the pre-RC in the beginning of G1 when CDK levels were low. Remember that was the origin recognition complex and this, now I'm blanking, the CDT1 and MCM, those proteins. So more proteins need to be added onto that complex in order for DNA replication to be able to occur. These proteins that are added to form the pre-IC need high levels of CDK or active CDK, I guess. So this would happen later in G1 once CDK is activated. And yeah, it says here that you don't need to memorize proteins for pre-IC formation, but you should know the ones for pre-RC. And then the last part of G1 is that the cell starts replicating DNA, and then that means that we enter S phase. And then that gets into next week's topics, which is going to cover the regulatory mechanisms of S, G2, and M phase. And you'll learn about the checkpoints in all of those and what happens in each of those phases more in detail. So that's all we had for today. You can stick around if you have any questions. Here are our emails and some fun science-y memes, I guess. Yeah, I'll stop sharing my screen.